To better understand how neurons work, we need to understand ions and how ions enter and exit cells. If you recall from the discussion on a plasma membranes are selective barriers. They allow certain molecules in, allow certain molecules out, and prevent a lot of molecules from just randomly entering and exiting the cell. Ions, because of their charge, either positive or negative, cannot penetrate through the plasma membrane. So they would require ion channels to allow that ion in or out. Ion channels are classified largely based on a few characteristics. One is what ion does the channel allow in or out, or what ions. Some ion channels allow only potassium. Some ion channels only allow calcium. Some ion channels allow sodium and potassium. So that's one feature that helps us characterize a channel. Coupled with that is the direction the ion is going. Typically, the ion is going to want to go wherever there's less of that ion, or it might want to go towards an opposite charge. If you're going to move an ion against a gradient, you might need energy. So we also need to understand whether that channel is an active channel, active transport, meaning it moves the ion against its concentration gradient, or is it a passive transporter, where that ion goes down its concentration gradient towards a lower amount of that ion. Then there is a stimulus quite often for the opening or closing of the channel. That's what this diagram is talking about. There's different ways to open a channel. One type of channel is known as a leak channel or non-gated. Leakage or leak channels, there's no stimulus that opens or closes the channel. The channel kind of opens and closes kind of randomly. So there's no specific stimulus to open the channel. There's a number of leakage or leak channels in a neuron. There are potassium leak channels, there are sodium leak channels. Rule of thumb is that the potassium leak channels are higher in amount. So there's more potassium that can leak in or out of a neuron than sodium. We're not going to be discussing leak channels very much. We're going to focus more on the gated channels. Panels B, C, and D are all types of gated channels which require a stimulus to open the gate, if you will. But depending on the type of stimulus, there are three categories of gated channels. There are ligand gated channels, there are mechanically gated channels, and there are voltage gated channels. A ligand gated channel will open when the ligand, or the chemical, attaches to the channel. So this purple channel here is a ligand gated channel, and the ligand is acetylcholine. We've heard about acetylcholine. It's a very common neurotransmitter. So this might be how a neurotransmitter is stimulating another neuron to transmit an action potential. Okay, so ligand-gated channel. In this diagram, what opens this red channel is a mechanical stimulus. Maybe there's pressure on this neuron, or there's a vibration, or there's a touch. Those are all mechanical stimuli which would open this channel. And then finally, there's voltage-gated channels. These are found along the axon. These are very important for action potential propagation down the axon. But we have to understand what the voltage means. Voltage is a charge difference between the inside and outside of a cell. And it's stating here that the voltage difference is negative 70 millivolts. That means that on this side of the membrane, there's a lot more negative ions. And negative 70 is actually going to be a really important number for us. That's the charge difference when neurons are resting. Okay, so when they're not an active neuron, they're typically kept inactive by this negative 70 millivolt charge. Notice what happens. Something caused that negative 70 to get to negative 50. So the voltage changed. And quite often when voltage changes, that will open a voltage-gated channel. And this is a voltage-gated channel for potassium. And we see that when this neuron reached the negative 50, potassium moved through. Okay, so I wouldn't be too concerned about what ions are moving through these channels. This is a figure just to teach us what we mean by leak channel, ligand gated, mechanically gated, and voltage gated. We're going to be using a lot of that terminology going forward. This is just a table to review what I just mentioned. It's discussing the 4 types of ion channels, a little bit of a description, and we'll be talking a lot more about their locations when we get to neuron function. But you'll notice that the ligand and the mechanically gated channels are most often found at the dendrites. So they're used at those dendrites when a neuron is getting stimulated, whereas the voltage gated channels are more common along the axons. So they're very important for the action potential. Next topic we're going to talk about is known as the resting membrane potential. I briefly mentioned it earlier. It's typically negative 70 millivolts. What that means is that there's a difference in charge that occurs at the opposing surfaces of the plasma membrane of cells. We often call this a polarized cell. So let's take a look at this panel down here. What we have is a plasma membrane. They're telling us that this is the inside of a cell, this is the outside of a cell. If we add up all the positives and negatives, a lot of them negate one another, right? So you might have equal number of charges, and then there would be a net 0. But there isn't. Notice that there's more positives on the outside of a cell than there is negatives. So there's a positive charge on the outside. The inside, there's more negative. So the negatives kind of line up along that surface. The positives line up along the outer surface. If we stick a probe, an electrode, through that plasma membrane, this electrode measures the difference between how many positives are here and how many negatives are on the inside. If they were equal, this voltmeter would read 0. But notice that the voltmeter is reading negative 70. That means that there's more negative ions on the inside here than there is positive ions on the outside. This is known as the resting membrane potential, which tells us that there is a net positive charge on the outside compared to a net negative charge on the inside. And the measure of that is negative 70 millivolts when a neuron is at rest. Okay, so this negative 70, you can think of it as keeping the neuron at rest, because we're going to see it changes, and when it changes, that triggers an action potential. I often think, and a lot of other people think of neurons as kind of like a gun. You've got a gun that you want off, so there's a little button called the safety. That makes sure that that gun is not going to fire. Okay? Then there's something that turns that safety off and allows the gun to fire. But in order to fire that gun, you got to pull the trigger. All right? So for our analogy, the negative 70 prevents the neuron from firing. Okay? Negative 70 millivolts. We often call this polarized because there's a difference. When two people are polarized, they have different views. In politics, Democrats and Republicans are polarized. They have different views on things. The North and South poles are different. They're on separate ends of the Earth. So that's why they're called poles. So this neuron is polarized because the inside is a lot different than the outside. The inside is negative, the outside is positive. Okay, so this negative 70 millivolts is the difference. This is what creates all the power for neurons. Neurons have to be polarized in order to eventually be stimulated to transmit an action potential. This is just another picture helping you visualize kind of the ion differences on the outside compared to the ion differences on the inside of neurons. Notice that on the outside of neurons, there happens to be more sodium and chloride. All right, there's a lot of sodium and chloride outside of cells. Those are very highly concentrated ions in the extracellular fluid. On the inside of cells, there happens to be more potassium. Potassium is an intracellular ion. There's more phosphate ions. There's more protein. So there's a difference in ions when you compare the outside and the inside of cell. Now, if again, a lot of these guys are going to negate one another, but notice that there happens to be more negative ions on the inside. And that's usually because of these proteins and these phosphate ions. They're negatively charged. On the outside, there just happens to be more positive ions. These could be those sodium ions on the outside. So we need to understand what the resting membrane potential is first in order to understand how a neuron works. The next video, I'm going to attempt to draw a neuron and explain how a neuron generates its action potential.